All right, welcome to what I like to call a BVEV, which stands for BBVEV, Brother Bailey's very entertaining videos. Um, Today we are going to talk about smell, and I want to keep it short and pretty concise. The one thing that I hope you recognize for all of these videos as we think about special senses is this. When we're talking about anything associated with senses and how to interpret those senses, we have to acknowledge that the brain only speaks one language, and that language is action potentials. So with all of these special senses units that we're talking about, the common goal is the same. How do we convert an external stimulus into an action potential? And that's what I want you to think about as we do this. All right, so here's our guy, and that's one of my favorite odorants. Is 3-methyl-indole. This is we could actually draw this thing out, not because it's important, but because it's entertaining. So this is 3-methyl-indole, and it goes by another name. It goes by the name of scatoli. You may recognize this from the root word scat, if you've heard that before. This is a common uh, additive in things like, say, ice cream, vanilla ice cream, strawberry ice cream. They'll put this in there to enhance the flavor because of its smell. You'll also find this a little bit more concentrated in high-end perfumes. The fascinating thing about this molecule is that if you recognize that root word scat, this is the same molecule associated with the classic smell of poop. And that's really interesting because they put it in ice cream, they put it in perfumes, and it smells like poop. Well, that's intriguing. Why? What's going on here? A lot of it has to do with the overall concentration of that odorant in the air, and that concentration will be processed in different ways. At mild concentrations, it's actually a pleasant smell. In more heavy concentrations, now we associate it with the smell of poop, something that's fairly nasty. So what's going to happen? This poop particle, this odorant, is actually going to go right up this guy's nose. And what's going to happen when it's in there? Well, if we look at what's going on inside this guy's head, we can add here's the nasal cavity. And what's happening in here? Well, what we're really interested in is this region right up in here. Of course, oh, I don't know what that is. That's interesting. Oh. The guy's brain is sitting right up in this region right here. This here in green, this is the location of smell. And we want to zoom in on that and see what this actually looks like. We'll start with the cribiform plate. If you've taken anatomy, you'll recognize this is part of the skull. It's actually a part of the skull that's perforated. Now, just to make sure we're all on the same page, this would be, in this drawing over here, this would be right here, okay? Where the skull itself is perforated, and we'll see why right now. Through these perforations, we actually have neurons that are extending their axonal projections. Okay, through the cribiform plate, now we can see why the perforations are there. We've got these cells that are extending their processes, and these are actually neurons. These are, we'll label these. This is an olfactory neuron. These are really, really fascinating because, well, collectively, they're called cranial nerve 1, the olfactory nerve. Individually, one of their unique superpowers is the fact that these things can regenerate, and they do so every 2 to 4 weeks. So, not as fast as, say, epithelial cells in the tongue when we're talking about taste buds, but the fact that a neuron is regenerating that fast is unique. And in fact, I know scientists have tried to take advantage of this. There was an experiment done in Poland several years back where a guy that was paralyzed from the waist down because of it was a bar fight, and he got stabbed in the back with a knife, severed his spinal cord. They used olfactory neurons from one side of his nose, and they took these neurons and transplanted them into this guy's spine. And because these neurons regenerate, they were able to go in and form synapses and bridge the gap and start closing the electrical connections in this guy's spine. And he was able to get some feeling back into his legs and start walking again over time as he went through rehab. So that's kind of cool. The fact that these things can regenerate is something pretty special. There's a couple other cells, these blue cells, not super important, but we'll highlight them anyway. This is called the Sustentacular Cell, and this is just a support cell, and it is epithelial in nature, just to kind of keep the area intact. And then the other cell that we have here is the green cell, and this is called Bowman's gland. Bowman's gland is important because these cells are responsible for mucus secretion. The reason mucus is important is because this is what the odorant gets dissolved in. So as we think about smell, we've got this poop particle, right? The scatoli that's coming into the nose. It's an airborne particle, and as it comes in, it gets into this mucus that's lining my respiratory cavity, and in the nasal cavity, it dissolves in this mucus. It becomes solubilized. And so this dissolves the odorant. And this is also important because with the constant production of this mucus, it's always, always, always just washing back down our throats. We don't recognize this, but often when we swallow, what we're actually swallowing is mucus that's coming from the nose, from the nasal cavity. And so that poop particle that you just smelled went in, dissolved in the mucus, and then you swallowed it after it registered its smell. So this is what washes the odorant away. And that's a good thing. When you have a bad smell in your nose, you don't want it to linger very long. You want to get rid of it. And this is how it happens, by that mucus just washing it all away. Now, let's zoom out just a little bit and let's see what's going on with these neurons, these olfactory neurons, because this is an important part of the business. So we have regions up here where they synapse. These synaptic areas here, here, and here, these are called glomeruli. So we will label this a glomerulus. Now, all of this that we're drawing constitutes a structure called the olfactory bulb. This is where the olfactory neurons touch base with and stimulate these mitral cells. These yellow cells here are mitral cells. And together, these mitral cells create what's called the olfactory tract. And where those go, that's an important thing to recognize. Remember, as we think about where that's going to go, first and foremost, they do not go to the thalamus, right? This is the one special sense that bypasses the thalamus. And we talked about reasons why that might be. Because smell is so important for survival of many species, including mating and reproduction, including identifying, locating food. This is a critical thing that's just hardwired specifically into the amygdala, hardwired into the limbic system to create emotional memory. And that's why smells can trigger such powerful memories and powerful responses, even in humans, because it doesn't go to the thalamus and therefore becomes fairly unprocessed directly into the limbic system. Now, if we're going to follow this destination eventually, where are we head up? Well, we're going to work our way to the olfactory cortex. This is on the ventral surface of your temporal lobe. From here, there's a couple of different places we can go. We can head out to the frontal lobe. This is going to allow interpretation. And this is what we were talking about before, down to the limbic system, specifically the amygdala, for some sort of emotional response. So there's smell. It's not that complicated. Kind of fun to see how all of this stuff works. I do want to take just a second to show how G protein coupled receptors work. I have a separate video for G protein coupled receptor signaling, so I'm not going to spend a lot of detail on this. I just want you to show what's happening here in the nose. So if we were to zoom in, say to this area right here, what would we find? This is where, on these olfactory neurons, this is where we're going to find the G protein coupled receptors. So let's label that. Location of G protein coupled receptors. Now, this is important to think about because it's estimated that even humans, I mean, you think, maybe our noses aren't that good, but humans, it's estimated, can distinguish between 1.7 trillion different smells. That's 1.7 times 10 to the 12th. It's a huge, huge number. How do we make those distinctions? You can see now how important this is. It's clear that over 900 genes in humans, this is over 3% of your entire genome, consists of G protein coupled receptors that are specific for smell. And the combination of odorants hitting these 900 plus different G protein coupled receptors is what gives you the ability to distinguish over 1.7 trillion different smells. Now, I'm not talking about sensitivity. Sensitivity, right, if we're talking about a dog being able to, track somebody down, sensitivity just comes with raw surface area. I'm talking, and of course, people with big noses, maybe you got a big schnoz, maybe you have increased sensitivity. For my part, I'm grateful I don't. There's a lot of things that my wife can smell, especially when she's pregnant, that I don't want to smell. She can walk over to a wall and say, there's a dead mouse behind that panel right there. Like, I don't want to be able to smell that. So I'm grateful that I don't smell very good. But the ability to distinguish between different smells, that's something we all have in common. And it comes from the fact that we have lots of these G protein coupled receptors. So if we were to zoom in on this, okay, what are we going to see? We have the surface of that olfactory neuron, and the odorant, of course, is coming in through this location and it's dissolved in the mucus, so it's solubilized. Let's see what happens. Well, we've got our G protein coupled receptor right here. And for any given one of these cells, they're specific for just one receptor. So, just as a recap from that movie that we watched about G protein coupled receptors, we know here comes our odorant. Here it is, our little 3-methyl indole, and it's going to bind, and that creates a shape change in the G protein coupled receptor itself that's going to activate alpha subunit. So here's alpha, and it is bound to a GDP, and because it's bound to a GDP, that allows beta gamma to tether it. Now, the job of the receptor, once it's activated, is to whip out its big pair of scissors, right? So out come the scissors. The GDP is kicked off, and we replace that with a nice new shiny GTP. So now we have an alpha subunit that is bound to GTP. And the whole purpose of that interaction is because when alpha is bound to GTP, Beta gamma can no longer bind to alpha and tether it. That liberates alpha and it's free to go do what it does. In this case, and again, if this is new to you, you should go back and watch the G protein coupled receptor video that we put together already. In this case, the target for alpha subunit is adenylate cyclase. Of course, we know adenylate cyclase has one job, and that is to take ATP and convert it to cyclic AMP or CAMP. Cyclic AMP in this scenario, cyclic AMP is going to go bind and open up a door that happens to be a cation channel This is going to allow cations, namely sodium and also calcium, into the cell. And of course, whenever cations are coming into the cell, what do you get? This is going to give us, we're talking about a neuron here, this is going to give us depolarization. So through the activation of a G protein coupled receptor, we were able to use cyclic AMP, our second messenger, to open up a cation channel. Introduction or entry of the cation into the cell causes depolarization, and that leads to an action potential. And voila, we just converted an odorant into an action potential, the language that the brain speaks and understands.